Good afternoon, everyone. Welcome to the Wednesday afternoon lecture. This afternoon, we're going to hear about Cryptococcus Neoformans from a distinguished investigator, Dr. Arturo Casadevall, who has just very recently moved from his previous position at Albert Einstein, where he has previously been for quite some time, about 22 years, and just now arrived about a month ago at Hopkins, where he is chair of the Department of Molecular Microbiology and Immunology in the Bloomberg School of Public Health. Arturo has a distinguished academic career, his undergraduate degree from Queens College, his PhD and MD from NYU. And as I mentioned, then taking up position at Albert Einstein and rising through the ranks to become a named professor and chair of the Department of Microbiology and Immunology there. He's an elected member of the American Association of Physicians and of the Institute of Medicine. And I must say, from my perspective as NIH director, I'd like to point out that he has also been a very dedicated servant when we have called upon him to help out with many different NIH tasks, including his current role as co-chair of the NIAID Board of Scientific Counselors, and his service previously on the NSABB, the National Science Advisory Board on Biosecurity. He has done important work over many years on fungal pathogenesis, but he also has some eclectic interests in other things, including the scientific enterprise, something about dinosaurs, concerns about research misconduct and how we are handling that very troubling issue in our community. And he's also currently editor-in-chief of MBio, which is the first open access general journal of the American Society of Microbiology, and one which has been publishing some pretty interesting stuff of late. So we're fortunate to have him here for this afternoon. So please join me in giving a warm welcome to Dr. Arturo Casadevall.
Thank you, Dr. Collins, for a very nice introduction. I'm going to be talking primarily about Cryptococcus Neoformans, but I warn you that, let's see how we go forward here. I'm sorry, here we go. That this is a seminar that is going to be all over the place. We're going to talk about Martians, Cryptococcus, publication ethics, the Chernobyl microbiome, fungi, capsules, intracellular pathogenesis, pigeon excreta, a special cockatoo, 2 movies, Melanin, and global warming. In the next 45 minutes, I promise you, it will all come together. In fact, one of the things that I rebelled against is that in today's science, they force you to be specialized, and they are not comfortable with you unless they can put you into a niche. Microbiologists, immunologists, cell biologists. I refuse. I consider that a scientific gulag, and I think you'll see that I'm all over the place. There is a lot that I will not talk about. I will not talk about antibody function, dinosaurs, humoral immunity, H5N1, gain of function, publication, retractions, impact factor mania, or enterotoxins, things that are also dear to my heart. So the problem of fungal pathogenesis, in essence, is, if you look at it from the big picture, there are too few fungal pathogens. In fact, fungal pathogens are not a big problem for most human populations that are immunologically well. The majority of fungal diseases continue to be caused by Candida, and it's acquired primarily from other hosts, in particular your mother, their mother's flies. From the environment, we get these other organisms where infection is frequent, disease is rare, and the outcome of most of the time is elimination or latency. But when there is disease, it is progressive, often lethal. Communicability is very rare, and these are 20th century primarily pathogens. In contrast to viral and bacterial diseases, most of these organisms were described in the early 20th century. So a brief history of Cryptococcus neoformans, discovered in the late 1900s by these two individuals, Otto Buse and Abraham Buschke. Abraham Buschke died in a concentration camp, tying this organism to one of the horrors of the 20th century. Between 1900 and 1950, only about 500 cases made it to the literature, but in the beginning of the 1950s, there is major progress in diagnosis and therapy. And here I want to celebrate the intramural program. I cannot imagine where we would have been in 1981 had the work that was done here in mycology had not been done. For example, we had a great diagnostic test. Dr. Bennett was involved in that. Jun Quan Chung worked out the sexual stage, not only for this organism, for many others. And by the 1980s to the present, it's become a major pathogen. Molecular tools were also beginning to be developed here in the intramural program. And today, this organism causes over 1 million deaths worldwide, the cases of which the majority of them become incurable and lead to death. Here is a portrait gallery. What is striking about this organism is this polysaccharide capsule, which is absolutely needed for virulence. And without it, the organism is essentially a virulent. In the big picture, infection is acquired from the environment. You have all been infected, whether it comes from pigeon excreta, desiccated cells, an arboreal source, we don't know. But in the majority of us, this is either cleared or it goes into a granuloma that may stay there for the rest of life. And later, hopefully it won't happen, but when people become immunocompromised by a variety of mechanisms, and one of the major, this is an area where there's a lot of activity going on now, because often normal people will come down with this and nobody knew why. Well, the group headed by Steve Holland is pretty much showing that some of these individuals develop almost like an autoimmune autodeficiency, by which they make antibodies, they knock off their own cytokines, and that completely disposes some of this disease. Again, the intramural program making a major contribution to this disease. Many years ago, we became interested about what would the relationship with pigeons. And this is the Bronx, and what you can see is the white circles are cases, the other places are contaminated sites in the environment. And we found that, in fact, they were indistinguishable, suggesting, but not proving, that in fact the disease is coming from local sources. It is possible that both sources are infected from a third. We also began to look at, we try to do a lot of work on serology and say, could we identify those who may go on to get disease, patients with AIDS? We couldn't. In fact, we could not find a single New Yorker who was negative in serology for HIV. So what good is a test where 100% of the people are infected? My suspicion was that it was telling us the right thing, but we needed to find a negative. And to do that, we went to children. And what we found was that children are born with maternal antibody, and then they become negative. But that the age of infection in childhood is somewhere between 2 and 4. And in fact, we think that this is one of the diseases of childhood that may not be currently undiagnosed. For example, patient 156 in the study showed up with vomiting to the emergency room and had an antigen titer of 1 to 16 confirmed. So clearly had cryptococcosis, probably resolved it. We had no way of following up because this was an anonymous type of study. But you know, this was published at a time, and this is what happens when you publish work like this. The New York Post ran this headline, and I will tell you something about human nature. David Goldman, who did the study, for the next few days, we had a lot of people call the lab, concerned parents, you know, my kid walked past the pigeon, should I do anything about it? You know, no, you reassure them. But the most interesting thing was what happened about 3 weeks later when we got hate mail accusing us to be bad scientists because we had not proven that the kids had not infected the pigeons. So the same piece of data was turned completely upside down. And I think that is the beauty of humanity, that no matter what piece of data you give some people, they basically can give you multiple interpretations. So a very special cockatoo taught us that you can be infected at home. This was a patient became progressively demented in Boston. She had had a renal transplant, and it was thought to be Alzheimer's, and after a while somebody did a spinal tap and it was cryptococcosis, and we were able to link the isolate in the cockatoo in the apartment to the patient. So how did I get to work on Cryptococcus? One of the things that I often wonder when I sit in the audience and people talk about their work is, how did they get here? And very seldom do they tell you. So I am going to tell you, I had no idea I would work on this. In fact, when I was a graduate student, that's what I looked like in the early 80s, I was interested in physical chemistry. I went to work with a physical chemist. I was interested in making measurements. You know, make the same measurement every day of your life and see how close you can do it and validate it. Look, we all have issues. Okay? We do. I like to do that. But I was very influenced by going across the street to Bellevue. And that's what I also looked like with those big glasses from the 1970s. And the AIDS epidemic was a formative experience in my life. I could not believe what I saw. I mean, I don't think that any of you, except if you lived through that time, could have a memory of what those days were like. Basically, The hospital was full of people, many younger than I was, and you couldn't do anything about it. These people just died. So I decided that I needed to get retrained, and I went to an immunology lab to work with Matty Scharf. And he put me to work in immunoglobulin mechanisms, to immunoglobulin genes, and I kept telling him, I really want to do something that I can do something clinically. And I went into the wards, and I took care of that man, a 30-something Cambodian man, with cryptococcal, and we gave him maybe 3 or 4 grams of amphotericin B, and it made no difference. Still died. And I took the isolate was sensitive. This was immune failure, and the question was, could you make antibody? I wanted to make antibody. Matty Scharf was an antibody lab, and so Matty wasn't too happy about this. In fact, he suggested that I talk to eminent immunologists, and I went to talk to 3 eminent immunologists, all 3 members of the National Academy, and this is the advice I got. You should work on relevant immunology, cell-mediated immunity. We know all about antibody, and this is what you're wasting your time. If you want a career, don't work on useful things. The more basic it is, the better you'll do. And get a different project. You're going to learn everything we have known about pneumococcus all over again. Well, I didn't take the advice. I continued to work on this. And 10 years later, we were able to do the clinical trial. One of those antibody monoclonals that I made actually went into clinical trial. And there are many people to thank, including Dennis Dixon, Richard Hafner, all the intramural, all the people at the NIAID. The phase 1 trial did show that you can reduce antigen. And then this compound entered into the valley of death, also because the problem was much lower than when I began, because of the development of highly retroactive therapy. And well, we have not given up on this. We have other ideas, but right now we are looking for cash to go forward. So in the 1990s, what happened? While this thing was being developed, I took heart the advice that I was given. Basically, stick to basic science. You can develop the antibody if you want, but get a basic science work on the basic problems. And in fact, we were able to do that successfully and found a lot of stuff in immunology that would not have been suspected from work with pneumococcus, which was then the poster child of an encapsulated organism. We found that the antibody responses were highly restricted, that antibody protection depended on isotype. One type of IgG worked, one didn't work. That if you take this IgG and you switch it to the other one, that you could protect. That antibody protection depended on specificity exquisitely. You moved it a little bit in the polysaccharide, it didn't work. Strangely, antibody protection required T cells. That's not supposed to happen. The constant region affected antibody specificity, basically going back and questioning some of the ideas that go back to the work of Porter, who got the Nobel Prize for basically showing that there were 2 parts to the antibody molecule. But the most important thing that came out, and I always go back to concepts, things that transcend your field, really are probably the most important thing you can contribute, was that you can take an organism for which immunology tell you that cell-mediated immunity was dominant and that antibody-mediated immunity had no role, and you could make a protective antibody. This was novel then. It is accepted now. And in fact, we went on to repeat this and to show we made the first protective antibody to tuberculosis, and now this has been repeated in many other areas. And this led to a rethinking of that old divisional labor that dominated much of immunological thought in the last 20th century. I don't think people talk anymore that way, that things are either one way or the other way. In fact, I think we now accept that this was from what should have been all along, that the immune system, an effective response, is an all-arms response. But by the late 1990s, we had a problem. And this forced me to go from immunology to basically work primarily in a cell biology. And the problem was that we could not explain the findings in immunology. Antibody-mediated protection was not due solely to providing opsonins, since both protective and non-protective antibodies were opsonic. So clearly, it wasn't that this capsule, once you overcome it, this will work. There was also the problem, why do you need T cells to protect in this situation? So one way to resolve the dilemma was to think that Cryptococcus may be an intracellular pathogen and that we were dealing with very different types of immune responses. So the late 90s question, is it? Well, this weekend was quite controversial. And you can see histology, this looks extracellular. If you get this is a granuloma from a child, it's intracellular. And if you do it in a mouse, you can go either way. You could basically argue that these are about to be phagocytosed, or maybe these are dead. So we try to do a lot of work, and I want to point out that Jack Bennett in 1973 had in fact noted that the organ, it grew much more rapidly in the inside cells than it did in serum, suggesting that something more was going on here in the pathogenesis. So by the early 2000s, this was established that in fact that this was a facultative intracellular pathogen. The latency involved persistent intracellular survival. It will hide out in your macrophages. It will live there for years. The mouse susceptibility was shown to be proportional to whether the macrophage was permissive or not. And most recently, and this was published in the last couple of years, the ability to replicate in macrophage correlates with lethality in humans. So we went from the unable to explain the immunology to basically rethinking very differently how this organism worked. So then you do the following. You go into the literature and you say, well, this is the type of strategies out there used by Listeria, Legionella, Chlamydia, Mycobacterial, Histo. And the problem is it did not match anything done by the others. So we were clearly dealing with something different. In fact, when we began to look at this, we found that this organism doesn't do anything at the time to the phagosome. It absorbs the hit. It goes into the macrophages and is able to take it. It takes the oxidative burst, it takes the proteins, and it's just not killed. Well, what's going on? The polysaccharide provides a tremendous amount of protection. Here is an experiment where you added some polysaccharide and basically exposed it to an oxidative burst. The polysaccharide capsule can absorb it. It also begins to damage the phagolysosomal membrane such that it doesn't able to keep the pH, and yet it resides there. It also has very powerful antioxidant systems, including lacase, and Peter Williamson has done very, very nice work to show that this stuff is secreted in here and in fact helps to deactivate the oxidative burst. So here is the problem. The problem also is that you have a humongous vacuole. The size of a bacterial vacuole will be 1,000-fold lower in volume. So whatever you're making is going to get diluted. It has to get through this capsule, then it has to get through the melanin to reach the innards of the organism. The organism will sit in the macrophage and simply survive. Then it also has an unusual thing in which it secretes vesicles filled with polysaccharide, and essentially the cell runs out of membrane. And it can promote later sort of an apoptotic kind of death. And by the circa 2003, we were pretty happy. You can draw a diagram, you can get featured in trends, but this was a very incomplete story, as I'm about to show you. So basically, there is no problem going in, it gets phagocytosed, you get a mature phagosome, and then the problems begin, these vesicles begin to be released, and at some time we thought this would lyse, and the cycle would contain itself. But the movies will show something different, and I'm about to show you this. I also want to point out to you that most of the findings in my lab are the result of serendipity. What you're going to see here is because somebody left the movie run. We run the movies for 2 to 4 hours. What happens if you run the movie overnight? Well, this is what happened. Here you see a macrophage, and you're going to see in a minute that it's got a bunch of cryptococcal inside, but something is going to happen here in which the macrophage is going to, the cryptococcals are going to come out around 8 hours. And there they go. They're going to go about now. And look at the cell. The cell in the back still left alive. And if you don't believe it, keep watching. It divides. And in fact, one of them will walk off the edge. So that's one outcome. Here is another outcome, the outcome that we thought we would see. This shows lytic exocytosis. These cells will burst. There it goes. And here is something that today remains unexplained, but it is a major problem in cell biology. You're going to see that a macrophage can transfer, look at that, it just transfers a crypto from one to the other. So the macrophage basically give it up, take it up, and this thing moves. And it's very frequent. I'm not showing you, I'm showing you events that are order of several percent. So we have been looking primarily at these two events to try to understand, because clearly from a cell biology, there has to be a lot of science to be found to understand this. And one of the problems today in science is if you get a movie like that, you can enjoy it for 5 minutes before somebody says to you, what's the mechanism, what's the mechanism, what's the mechanism? And if you give them the mechanism, then they say, does it happen in vivo? Does it happen in vivo? Does it happen in vivo? The bottom line is, you're scientists. We should be able to enjoy the movie. In fact, what's important is the movie. I can think of 10, 20 mechanisms by which this can happen. You could have snares. You could have, fusogenic membranes in other ways. So, but we live in the reality of today, and if you're going to work on this, you got to answer those two questions, and then you're free to go on to wherever you're going to go. So, bottom line, it does happen in vivo, and it happens in 50% of the macrophages. So it's a very, very common phenomenon. The mechanism is incredibly complex, and I'm not going to go through this because this is a general talk, but I'm going to show you that these things, well, what I want to show you, this is one of the movies. Here you have the opposite of this. I don't know how many of you have seen the movie Alien, but the alien has the same problem as Cryptococcus. It wants to get out. In this case, though, it does a tremendous amount of damage to its host. But I'm going to show you what happens here. This makes giant autophagosome. You're going to see the LC3 all stained on the surface by the time this ends. So this is a very, very sophisticated process. So at some point, this thing will now fuse, and these things will come out. Turns out that actin rearrangement, pH, annexins, autophagy, all these things are intricately associated, and none of them is all or none. You're talking about redundancy in this system. So one of the things that we've been focusing on is the phagosome. And in fact, this leaky phagosome determines the outcome of what happens. The cell, even though it's alive and will live for the next 2 or 3 days, has been hit and will die. It's undergoing a tremendous amount of damage. But that damage depends on how leaky the phagosome is. If you, for example, will make it more leaky through the addition of aniline, you can promote all lytic exocytosis. Michael Davis, who I heard may be coming to this talk, and Michael Olszewski have a beautiful paper that is currently in press, and they show that, in fact, things like gamma interferon that inhibit exocytosis, what they do is they maintain the integrity of the phagosome, and if you can do that, you can pretty much change the outcome of infection. So these macrophages are sustaining damage on many cellular systems. Their mitochondria are depolarized. The caspases have been activated, but they are not being activated at a level that is leading to necrosis, apoptosis. It's low-level damage incurred. Why? Because the organism can replicate much faster as long as it's inside. And what it is able to do is it's able to take care of the cell, and it is not necessarily in its interest to get out. And this is a paper that is now in press documenting this phenomenon. It turns out that the Cryptococcus is secreting large numbers of cryptococcal proteins into the cytoplasm. And without going into this, what you can see is that if you put live or dead cells, very, very different cells, proteins that recover from the cytoplasm. In this case, they are the result of digestion. In this case, they are the result of actual secretion. What I'm getting at is, I want you to think about the big picture and not focus into the... This is an organism that lives in the soil. It has absolutely no need to do any of this. Why is it doing it? To me, that is a very compelling question. Because what you have is, you have great sophistication. This organism is keeping the cell alive, it's damaging the membranes, it's activating caspases, it's depolarizing the mitochondria, and yet it is not killing it because it is replicating. And this thing can get into insects, reptiles, worms, mammals, and even plants. So you have sophistication and non-specificity. How do you reconcile that? Generally, when we see highly sophisticated viral strategies, we say, oh, they co-evolved. This is a mechanism for it to maintain itself. This is the complete opposite. This is doing to hosts things that it doesn't even need to do. If you remove all the animals from the earth, its organism will probably still be fine wherever it is in the environment. Well, we began to think that you have to go back into the environment to think about this. And this is incidentally the back of a New York City hospital in 1992, and you can see how much material there was. So you ask the question, what is going on here? And we answered, could the common denominator be amoeba? Amoeba were known to be predators, and then there was at the time the Legionella story. This is around 1998. So we set out to look at the problem, and immediately we saw everything that we were seeing in a macrophage could pretty much be replicated in amoeba. You could see the phagosomal damage, you could see the formation of the vesicles, and this began to suggest the Cryptococcus had learned to be a pathogen, not because it ever cared about a mammal, but rather to survive predators in the soil. When you look at the genes that are regulated, you find that in macrophages, they're almost identical. When it's in a macrophage and amoeba, it's dealing with the same problem. It's dealing with something that ate it, it's in a big phagosome, and it's having to deal with oxidative bursts and microbicidal proteins. So another example of this is relevant. You can take a virulent strain and passage it through dictyostelium and recover passage after only 4 days, suggesting, in fact, that some of these things may be epigenetic, because I cannot imagine genetic changes. So you get to all this phagocytosis, both systems, intracellular replication, both systems. Both are toxic to amoeba and to macrophages. Non-lytic exocytosis, that phenomenon I showed you, can be demonstrated in amoeba. Phospholipid-triggered capsule enlargement, I'll show that in a minute. And the capsule protect against both. You basically have virulent factor concordance against two hosts that are separated by more than 1 billion years of evolution. Now this idea has been extended to many other fungi. So when you think about those organisms that come out of the soil, the aspergillus down here, things like Paracoxy, or others, Histoplasma, Blastomyces, why are they virulent? They're coming at you already with ready-made virulence. This has been selected in the environment by mechanisms that had nothing to do, probably, with any of the animals. In fact, non-lytic exocytosis are now being demonstrated in Drosophila. It's been demonstrated in amoeba, and it's been demonstrated in human and mouse cells. When this organism gets into any of these phagocytic cells, it has the redundancy to deal with processes that are separate by 1 billion years in evolution. So what do we think is going on? A lot is going on in the soils, and these things are being generated stochastically. And occasionally they may cycle through animals. And then what we see as a human pathogen is that occasional one where either there's been a breakdown in immunity, it cannot be contained, and then it appears to us as an organism that is quite deadly. The two main virulence factors are the capsule and the ability to make melanin. So, in fact, the capsule contributes for about 26% of the total virulent composite, and melanin for 14%. To my knowledge, this is the only organism for which this type of analysis has been made. Many people work on a virulence factor, and they love it, because if they knock it out, the organism becomes avirulent. But that could be contributing 1% of the total virulence composite. And in fact, what I urge everyone who's interested in pathogenesis is to try to do this type of analysis and to try to sort out what's important. And the fact is, our experience is consistent with it. If you give antibodies, you are able to neutralize that capsule, you're able to in fact, neutralize virulence. So one of the fascinating things about this organism is that it is able to rapidly enlarge the capsule. So you take a culture, you put it into a mouse, 18 hours later, the cells are much larger. You take the same thing, you throw amoeba in there, and you also get the same phenomenon. However, these cells can now become very, very large. This size is about 50 microns. This forms a formidable problem for the immune system. And these cells cannot be phagocytosed. The best you could do is form a giant cell around it. In fact, if they were colored, you could probably see them with the naked eye. One of the things that we worked out is working from amoeba now, we're able to walk backwards and see what this sensor is. So what happens is that Cryptococcus is secreting things like phospholipase B and proteases, and they begin to cause damage on the amoeba. And the amoeba then begins to release phospholipases that then triggered the capsule to enlarge. So this is almost like a danger sensor for it. If you sense phospholipase around, there is probably a predator. And in fact, the same mechanism is used for both amoeba and macrophages. So we went on to work out this quote-unquote mechanism. So it happens to be fission, duplication without fission. These cells become larger. They go N, 2N, 4N, 8N, and they don't divide. And in fact, what they do is the phagosome, to maintain the problem. The reason you cannot keep a cell growing is because the ratio between volume and surface grows disproportionately in the volume area. But the organism solves the problem by making a very large vacuole that squeezes the cytoplasm on the surface so that you again have a surface that you can work with. So I want to tell you a little history here. It's 2008, it's the summer. I'm in a Gordon conference. I'm in the back of the room answering emails. Why? Because I live to answer emails. And in fact, I have mastered the capacity to listen to talks and answer emails at the same time. And I'm listening, and a young scientist who's an assistant professor at the time is presenting. And I am shocked because she's discovered the same thing. And she has the same data. And I ask her the question, what do you think I did? I could have taken my cell phone out, going outside, called up the postdoc and said, you wouldn't believe, you're about to get scooped. You better start showing up every weekend. You're going to have no career. You got to get this out. Right? That's one. You could have also walked around the meeting and said nothing. Or you could have approached the scientists and said, you know, we have the same data. Let's try not to scoop each other. Let's try to get the best work done. At first, I think Kirsten was a little suspicious because, you know, I have a big lab. She was just starting out. This is the major finding in her lab. To me, this is just one of the many things I'm doing. But you know what? We began to get together, and we began to work together, and we waited for the other one to complete the experiments. And then we submitted together the papers, and they were published at the same time. Nobody gets scooped. Science is confirmed at the same time, and the work gets to a higher level of completion than if we had basically gone out there and try to scoop each other. So that raises a question. Why do you care? The reason you care is because our economic system in science is based on priority. So what is the money in science? It's prestige. How do you get prestige? You get priority. It can be related to credit. And in fact, you can use prestige. You get prizes, grants, publication venue, and then it basically results in more prestige. This is our economic system. This is how we do science. And I ask you, is this the best way to do it? Well, we have written this. In fact, science follows a winner-take-all economy. And I argue that we should rethink it in many ways because there may be better ways that we can do this. Why, for example, competing labs not wait for the other one to compete and publish at the same time? What if we gave credit to groups that cooperated and produced better data that was reproducible rather than groups that published first? I will tell you that I have done this 4 times, and every time it's been a wonderful experience, and it's right here. I did it with Robin May when we published the non-lytic exocytosis. This is the example I just told you about, the cell-to-cell transfer, and we have just done it with Michelle Olsasky and Michael Davis. These papers are in press. It's wonderful, and I will tell you an anecdote on it. The system tries to defeat this. It turns out that one of these sets, I'm not going to get into it, went to a high-profile journal, and where the editor gets no gain by publishing 2 papers that have the same thing. Why? Because the editor is concerned about the impact factor. So if he gets 2 papers on the same thing is divided by 2. The editor tried to cut a deal with the other group to only publish the other paper. And because trust had been established and we were together, the other group pulled out. And then we went to another journal. And I think this is what I would argue, if I can leave you with one thing of this talk is, if you think you're going to get scooped, if you have a situation like that in your lab, pick up the phone and try to see if you can work something out, do it together, share the data later so you can come at it independently, but you can end up benefiting. There are no losers. You end up with a much better relationship, science benefits, and at the end of the day, everybody is a winner, including society that doesn't have to deal with the reproducible results. So I've written a bit about this. This is in press and may have been published. And one of the things about competition is, when people think, well, you need to compete in order to promote people in science. You know, the fact is, there's no evidence for that. One of the transformative discoveries are made often in the absence of any competition. When you create competition, what you do is you often create people to solve the problems by the means that they know how to do it. And you stifle creativity. And there is some degree about that. And we should think very hard as we go forward on whether there are ways in which we can do science better. I want to tell you about melanins, the world of melanins. So we cover ethics. That was the 2 lines in ethics. Melanins are made by all biological kingdoms, including mushrooms and the skin. Cryptococcus makes the same type of melanin in your skin. It's called L-dopa melanin. This stuff is put on the cell wall, and it pretty much serves as a barrier, as a non-specific barrier that can protect it against practically anything you can throw at it, from antifungals, so this could be a cause of acquired drug resistance, heavy metals, defensins, your favorite oxidant, go on and on. So one of the things that's fascinating about melanin is that it is totally resistant to boiling in hydrochloric acid. So if you take melanized cells and you do this, you can recover melanin ghosts. And this has been an enormously useful procedure that has allowed us to study this. I want to point out the structure of melanin is unknown. Why? Because it is an amorphous compound. And basically our science is very good at solving structures of things that can go into solution or can be crystallized. Therefore, this is beyond our scientific horizon. But one of the things we have done, this is with atomic force microscopy, you can see that it is composed of very small things, and we think that it is made in vesicles and shipped to the cell wall where it's packaged. But I'm going to tell you about two melanin tails from the far side. So they have in common some things. First, initial incredulity, a collaboration with a long-term collaborator called Kate Darachova. They both involve radiation. Terrible difficulty publishing them. There is difficulty in funding. In fact, none of them have been ever funded to date. They've been confirmed by other groups, and they are potentially far-reaching. And this is why I always advise that you take some part of your day and read outside your area and try to make a link between some crazy thing and something that you're doing, because science will be a lot more fun that way than basically reading always or always sticking to your field. So tale one begins with Chernobyl disaster, 1986, molten Chernobyl disaster. Somebody sends in this robot into it. This is the only picture of it. And you see this wall back here is black. The reason this wall back here is black, this is the molten reactor area, is because fungi are growing on it. Has the good idea to culture it and find that melanotic fungi are growing all around an area where if you walk in, you are lethally irradiated in something like 5 minutes. This gets published in a journal with an impact factor less than 2. So we began to think, could the answer be radiation shielding by melanin? So Kate Darachova sets up classical radiation shielding experiments. You take a 96 low-tech, 96 well plate, you put a film under it, and you do a radiation. So it turns out that melanin appears to provide quite a lot of, melanin alone gives you twice the amount half the amount of protection as lead. Think about that for a minute. This is a carbon-based substance, and you're getting half of it of what lead gives you. So if you double the size of it, it begins to function almost as lead. So again, that gets published, and, but that could not be the explanation. Why? Because fungi are remarkably resistant to radiation. In fact, the fungi wouldn't even care about the fluxes in there. So then we get another hint from, again, another not well-known journal, in that fungi tend to develop a phenomenon called as radiotropism, that they tend to grow towards radiation sources. Boy, isn't that interesting? Think about it. Why would a fungus grow to a radiation source? So we began to put two and two together and ask the question, could the fungus be using radiation for growth? And without going through all the data, it is published. It's now published in an open access journal. You can download if you're interested. The answer seems to be yes. Basically, radiation charges up melanin. Melanin can convert NAD to NADH. We think that this is an oxidation reduction loop that allows it to do. This paper, by the way, those of you who heard my lecture last year, I told you the gory details of trying to publish this. 3 years dealing with single-word journals. In the end, we didn't succeed, but we ended up in +1, and I'm very happy about it because it's the same data had been published in the other place. And the same people that turned us down had to write stories about it. In fact, I often ask people, would you rather have your paper in the reports or in the news? And you should always have to take the news because these journals have very high retraction rates. So news articles don't tend to get retracted. There was one criticism of the work that bothered me, and it came from a physicist that says, Arturo, gamma rays go through everything. What are you talking about? And we began to spend the next 5 to 6 years to show that, in fact, gamma rays could change the structure of melanin, so it had to interact. But that was not the proof that I wanted. The proof that I wanted came from this individual who basically created a melanin electrode and then irradiated the melanin electrode, and guess what he got? He got a current. There is no way out of it. Melanin has the capacity to capture this material by ways in which we don't understand and turn a current. If that individual can measure a current, he too published it in a single-word journal, but not one of the other ones. This means that biologically, this could in fact be used. It's now been confirmed by another group, so it is out there. And we have extended it to the visible spectrum. Blue light does apparently the same thing. And this is a picture I took on the summer, and you can see what I think is happening is the melanotic organisms are in fact probably having some form of photosynthesis that is complementary and possibly different than the one that is going on by chlorophyll base. And this is something I saw in Central Park this summer, and I cannot believe that these organisms are not harvesting sunlight with their melanin. These are using chlorophyll. These are using a very, very different system, I believe. And I think so this is something very different that came out of working and being interested in linking things differently. The Black Mountains of Antarctica. What's going on here? The reason they're black is because they got lichens growing on it that are melanized. Where are they getting their energy? I think they're getting their energy from the sun through a melanin-based photosynthesis process. The space station is contaminated with fungi. Little discussed issue. Here you see melanotic fungi growing they grow, by the way, very well in space, also consistent with growth in high radiation environments. So the tail, or the second tail from the far side is, remember I told you, I show you the data, that melanin has roughly half the shielding capacity as lead. Could we use it to protect mice against lethal radiation? The experiment worked incredibly well. You basically take mice and you feed them black mushrooms, or white mushrooms from synthetic melanin, and you can protect the mouse against a lethal radiation dose. What is going on here? Well, it's complicated, but I think this is what's happening. The melanin is providing local shielding to the gut, and then the gut is allowing to do an autotransplant so that the mice are able then to repopulate themselves. We took this data and we sent it to Japan during the Fukushima. I don't know if they did anything about it, but this is a low-tech, incredibly effective way to shield, to do some radiation protection. Now you say to me, well, I don't believe it. Well, it's been confirmed by another group, and they have the same data. Basically, you give melanin to mice, you irradiate it, you can get 100% survival. One of the crazy things about today's science is that if you came up with a small molecule that went into one of the signaling pathways and you got 20% thing, people would think it's a breakthrough. If you tell them black mushrooms will protect the mouse against radiation, I don't know what that means. It's published. I'm going to tell you one more tale, but this is not very different. So from one of the things is from ghosts, we made monoclonal antibodies, and we're able to show that melanization occurs in vivo. So we had an idea, could these antibodies be used to treat melanoma? So here's skin melanoma, and people say, it could never work because melanin is an intracellular pigment. It will never reach it. But you and I know that any tumor is mostly dead cells. And in fact, when you stain it, you can see that the antibodies will stay melanin. So the idea was, could you take the antibodies to fungal melanin, radiolabel them, and use to treat melanoma? So we did this in mice, published this in 2004 in PNAS. It worked beautifully. In fact, the melanomas are become essentially scar tissue, and this was then taken into humans, and it's been given to about 22 patients. And this project is currently stalled. We're trying to get it back. Again, it's in the valley of death. But we think that this is a potential therapy against melanoma that will basically add to some of the things that are already an area where there's been a lot of development. But I would point out to you that how these discovery come in, it came from a very, very different angle that was not at all related to cancer research. So as we go forward here, in the next 5 minutes or so, I want to give you a bigger picture. So you have soils, and in soils you have a lot of organisms. And they are all under selection pressure by rain, sun, amoeba, tractors, etc. And obviously, they all tend to survive. And most of the soil organisms must have the capacity to survive predators. Otherwise, they wouldn't be there. But only a few of them are virulent. What is the mechanism? Well, when you begin to look at the soils, you find an enormous, all the virulence factors are there. Whether you go into the bottom of the ocean, they're there. And we'll begin to think that the microbes were in fact playing cards in some ways. That is a random game, is what you get. So the mammalian hand must have the ability to grow at 37 degrees. The plant hand doesn't need that because plants are room temperature. You can imagine the insect hand doesn't need the same thing. This is a very useful way to think how these things may emerge. And then I realized that this had already been described, and it was described at the end of the 19th century by a Martian hand. And what happened then, if you read War of the Worlds, is that the Martians invaded. They beat up the London Police Department. And after being here a while, they basically got sick. They had never been to the planet before, yet there were pathogenic organisms that could take out the Martians. The lesson probably is that the more diversity of microbial life, you will always have some degree of pathogens, and they may just be emerging by the random combination of virulence factors. So one of the things that we are protected, we go back, why are there so few fungal pathogens? Well, we have adaptive immunity, but that cannot be the answer because things like frogs are very susceptible to it. What happens to be is that the majority of the fungi they lose viability very rapidly in the range of between 30 and 40 degrees. And in fact, if you take this data, that is the benefit of being hot, and you took the cost, and you solve these two simultaneous equations, look the temperature that pops out, your temperature. And in the past, the lecturer, when we talk about the dinosaurs, basically argue that perhaps the reason that there are so few fungal diseases is because fungals were the filter by which we got here. Mammals make no sense without something that allow them to compete with the reptiles. So to the end, the world is getting warmer, and it turns out that most fungal diseases currently come from the tropics. Makes sense. And that is an area where the fungi will adapt to higher temperatures. So all projections are the globe is getting warmer. And the question is how warmer it would get, and that is where the disagreements are. So we have looked at the phylogeny of thermal tolerance. Can we identify where the next pathogens may come from? I will point out to you that most of the organisms out there in the fungal kingdom are not susceptible to amphotericin, not susceptible to any of our own drugs. They're being protected by this temperature difference and our immune system. The fungi can adapt very rapidly to temperatures. Here is an experiment that was done by a company in Florida. They have a fungus, and this fungus they want to use to kill insects. The insects learn to sun themselves, and by sunning themselves, they kill the fungus. So they adapted the fungus within 6 weeks to be able to grow at 37 degrees. The message is that these things are rapidly adapting, and we in fact have unpublished data that is currently being reviewed, showing that if you take the average of thousands of fungal strains deposited, there are upward trends for the Basidiomyces, not the Ascomyces, suggesting that this process is already happening as the globe is beginning to warm. So could we predict anything based on what we know? Well, we went to a very simple system. We took Cryptococcus, we know a lot of it. We went to close relatives. This is work that was done here at the NIH by Jun Kwan Chung, in which he got the closest, and we took 3 of them that were relatives, but we had to go to a host where we could compare infectivity. And you had to go to Galeria, you cannot do the experiments in a mouse. So remember, the capsule and melanin gives you half of your virulence. So you would have predicted on the basis of this, here you have Cryptococcus, it kills all the Galeria. This organism that had no capsule and no melanin was more virulent than one that had capsule and melanin. Presumably, the hand of this organism included cards that allow them to take this thing. So it's consistent with the concept of emergence. And the question is, will immunology and infectious disease ever become a predictive science? If you know all about the immune system, if you know what's out there, could you ever predict whether things are in fact going to be pathogenic or not? The concept of emergency goes back to the ancient Greeks. The whole is greater than the parts. The fact is that if you know all about the laws of physics, you know about Bernoulli's equations of flow, and you know about the physics of grain, you cannot predict this. You can certainly not predict this feature over here. You may be able to explain it afterwards. But this suggests that there may be epistemological limits to what we can know in the systems. So the consequences of virulence as an emergent property is that it is not reducible. And this is actually a very scary proposition, because microbiologists basically keep the host constant and torture the microbe. Immunologists keep the microbe constant and torture the host. And they both then make conclusions and write papers. And what I think we're going to need new approaches to begin to think how these entities interact. And I think that I am optimistic. I think that we may get to something similar to weather prediction, in which you may be able to say with some probability, Arturo Casadevall, genetics and background, would have given him a high probability of getting the flu this year. And that's why I'm hoarse, a couple of weeks ago. So, but we may never get to a point that we may have a absolute prediction. And infectious disease will always be with us and will always be a threat. And I'm just showing you that the environment is probably the greatest threat that we face. So at the end, some insights came from this work. Non-specific environmental pathogens could have very sophisticated strategies. Things in the soil could take down your macrophages by a way you may not even have imagined. Virulence can emerge through selections independent of the final host. Melanin is a natural radioprotector and an energy harvesting molecule. This idea, by the way, that the fungi or melanotic organisms may use radiation for food changes how the ecology of the earth, because we think of the fungi as digesters. But if they are also producing energy by harvesting radiation from the sun and things like that, you have to recalculate the energy flows in the planet. Human mammalian temperature restrict most fungal pathogens, and global warming may breed you new fungal diseases. Thank you very much.
Well, thanks, Arturo, for a delightful romp through a whole host of interesting issues. We are open for questions. People who have questions, please come to the microphone so people on the web can hear you. And yes, please, go ahead.
Hi, Arturo. Very nice talk. So I have a question. What do you think, and I include myself on this, but why do scientists have such a difficult time identifying and recognizing innovation, especially when we're within committees? And do you know of any systematic study of such a thing?
This is a central problem. The director is right here, and this is something we need to solve. Basically, the way we try to recognize innovation is we give it to established scientists to look at. They're working from the current model. It's very, very hard for them, and the more it challenges the current model, the more likely it is to be rejected. This is the history of science. And I think what we need to do is we need to be more open-minded. And instead of rejecting something like this, says to you, this is the data you got to give me to believe it. And change the question around. And I question how much innovation are we just killing? Simply killing by the fact that we know what safe science is. We know what we need to convince. This is not the kind of thing you would ever put in a study section grant.
Let me add to that. So I think another area where one often sees innovation, if you're just looking sort of historically and retrospectively, is when you have two different disciplines coming to work together on a problem where they haven't previously partnered before, and now each one brings a perspective to that problem that neither one alone could have achieved. Do any of these examples fit that, or is that something different?
Thank you, Dr. Collins. They do fit that. I mean, I do come from a physical-chemical background, and I think differently when I approach the problem. I don't think as a geneticist, I think as a physical chemist. But Dr. Collins brings up something fascinating, and it's another problem. We think that great progress is made in the interfaces, but fields kill the interfaces. Think about it. They want you to be a microbiologist or a cell biologist or an immunologist. Try to change fields and see what will happen to you. Your old field will, they basically say you abandon them, and the new field will work very hard to keep you out. What is going on here? When we know very well that this is exactly what we should be encouraging. The fields work as social units that keep information either out or something, and then everybody says, we want more people to be interested in my field, but do they welcome anybody? Think about that. Do they welcome anybody? And first thing you do, your first paper will be tortured to death over some minor thing. You didn't use the standard strain in the field, or you used a pH that was 0.02 from what everybody likes. This is crazy. We need to fight this. We need to, when anybody comes up with, in your field, they come in and don't say, well, that's an entomologist. I don't know what they're doing. You say, let's see what we think is right about what is going on and embrace it. And if we do that, we could push science so much farther than if we basically try to squelch it every time we run into it. Yes, sir.
Well, I was intrigued with your comments on the melanin effect of radiation protection. Department of Energy spent millions of dollars to develop radioprotectants. I was wondering, did you find any application in that area?
We have applied for funding and things like that. And listen, the idea that something that melanin is going to protect, it simply hasn't connected. The data is there. It's been confirmed by another lab. The experiments are very simple, and I invite anyone to do the experiment. It's remarkable. This thing is protecting the gut, and by protecting the gut, the animals survive.
Is it any specific type of radiation sensitive?
Yeah, these animals, I forgot, they were blasted with usually the one that ablazed the bone marrow. These animals would have died, they died in about 14 days of sepsis and complete, and the gut leaks. So another mechanism that is probably working is protecting the gut locally so that you don't have the leakage that causes the death.
Thank you.
The timetable, surely there must be some window where you have to apply the melanin and wait a certain period and then zap with the radiation. How does that work? Because it must not be a long-term effect.
You're right. I mean, it's as long as it's in the gut. The melanin is not absorbed. It goes right through. It sits there. And what I think is happening is those little melanin granules are sitting down and they're providing local protection. And that local protection is enough for you to bounce back.
Let me also ask about the strains that have the melanin, which seem to love radiation, and it does make you wonder in terms of the evolutionary effect. Why would these particular fungi have been exposed to those levels of radiation during evolutionary time? Or is it just that they have a system that doesn't particularly care about the wavelength and anything will do?
That's actually a very insightful question that we have thought a lot about. Melanin is very cheap to make. All you need is to polymerize it. It is a pigment that is found in all biological kingdoms. The early Earth was a very radioactive place. It's probably a pigment from the early days that has got many, many functions and has been adapted for many, many uses. And I think that it's probably a low-tech energy absorption mechanism. Look, it stands to reason. Is biology going to waste things that are being answered? Come on. These things live off caves, electrons. In biology, whenever you can find an electron, it tends to use it. So my own view is that this is a remnant from a very distant time when there was no oxygen, the world was very different, and life was very simple. Yes, sir.
Thank you very much, Dr. Casadevall. A very interesting talk. I've read some things about your proposal with Lisa Perovsky on the host damage response network.
Right.
And you didn't quite touch on that. I was wondering, cryptococcosis in the non-HIV patient, you had shown in some of your work very elegant pictures of depicting from a commensal to disease process. Where you see that currently in what is known about cryptococcosis in the non-HIV
you're talking about
host.
the U-curve. So I think
Correct.
you get into trouble at either end. I didn't get into all the data. So at the low end of immunity, the damage framework is a theoretical construct that we proposed 15 years ago to try to reconcile the host and the microbe in microbial pathogenesis. And it's a very useful tool to think about it. In weak immunity, you get into trouble because the organism replicates, and this is what you see in AIDS, meningoencephalitis. At the other end, you get into trouble too. Now we see it with iris. And we now see evidence, at least there is evidence, association, not causation, that this organism may be related to asthma, especially in the inner cities, consistent with the hygiene hypothesis. All these kids are getting infected. Their lungs are all being polarized towards Th2. You remove all the diseases that used to give you Th1, but we're still letting them get infected with Cryptococcus. So I think there is trouble at either end.
Thank you.
Yeah, last one.
Thank you for the talk, very interesting. My question is, I like your example of the cards chosen by the organism. If you have the cards chosen by Cryptococcus gattii that infects immunocompetent versus the ones chosen by Neoformans.
You mean between gattii and Neoformans? I mean, I think that we don't know enough there to kind of pick out, but they're clearly different in that some of them, one of them is associated with immunodeficiency. I think that comparison is going to be very, very interesting. Thank you very much. Thank you.
And for those of you interested, there will be a reception in the NIH library right down the hall. Please come in and continue the conversation with the speaker if you'd like to ask more questions. Thank you.